Oh. Hello everyone, yes, you join me for the next instalment in this acquisition of behaviour series, and today we're going to move on to something slightly different. Now remember in the first video when we talked about cognition, we defined what it meant and what kind of things it included. From now on we're going to start looking at the individual fundamental mechanisms for cognition. And today, like it says on the board, we're going to focus on perception and attention. Now, the environment is full of loads of stuff, isn't it? Which organisms have to respond to. And this is very important for their survival, but every animal perceives its environment in a very different way. If we talk about vision, for example, us humans are trichromatic. So we have three peaks across our visual spectrum, seeing the three primary colours. But if you look at things like birds, starling for example, they're tetrachromatic. So they see in four colours. Okay? Obviously our hearing. Differences in our hearing between different animals. I mean, that's pretty obvious when you compare our hearing to that of a bat. For example, we hear in completely different frequencies. What we perceive can even change with age. For example, as you get older, your ability to see bright colours decreases. And that kind of explains, I guess, the stereotype of old age people wearing brighter coloured clothes. You can debate how true that is, obviously. Now our senses need to be optimised for the environment that we live in. If you take a British woodland, for example, the different light intensities occur at different parts of the wood. So depending on where your niche is, you need to be tuned in to the right sensitivity to help you survive. Now, as you might imagine, determining perception in different animals is quite tricky, but there are two main ways we can go about it. The first is using signal detection theory, and the other is sort of psychophysical experiments, but we're going to go into them a little bit later. First, we need to actually properly define what we mean by perception. And I've written them on the board here. I know there's a lot of words on the board today, I do apologise, but they're not saying that much, if I'm honest with you. So the definition of perception is the process by which an organism detects and interprets information from the external world by means of the sensory receptors. Okay, so that makes sense. The two key words, I guess, are detects and interprets. So it's how we process information that we receive. Okay, and that will eventually lead to some sort of response. Okay? Now there are certain principles that we need to take into account when doing particularly psychophysical experiments, which we're going to go into later. And there's 5 principles here. First thing is that species differ in what they perceive. We know that. We'll look into that in more detail when looking at bees and how they can see in UV light. More on that later. Species differ in their ability to perceive stimuli of different intensities. Okay. Sensory neurons respond to stimuli of greater intensity. Obviously, a lot of neurobiology does tie into this, but that's a completely different subject altogether because neuroscience is neuroscience and behaviour biology is behaviour biology. So we're not going to touch on the neuro side of it. You'll be thankful to know. Senses habituate to prolonged unchanging stimuli. That's also sort of neuro-related. But we're going to be talking about habituation in more detail in a later video. And responses depend on the environment which you're in, which is quite obvious really, isn't it? So usually when we're studying behaviour, we want to know why an animal is behaving in a particular ecological context. And an example which we've been using loads and loads, and we will continue to use throughout this series, is birdsong. Okay? And we know that birds are using birdsong to attract a mate, or to choose the right individual in which to mate with. Okay? Now obviously, there's loads of costs if this bird made the wrong choice. So poor genetics in the offspring would be obvious. It'd be a waste of energy, especially if it mated with the wrong species. I mean, that'd be kind of embarrassing, quite frankly. It could increase your chance of predation, or you could die as a result in some extreme circumstances. Now, to work out how animals respond to a particular signal they're given, we use this thing called signal detection theory, which isn't just applied in biology, it's also applied in many mechanical systems, but that's beyond me, quite frankly. And the whole idea behind signal detection theory is that the world is quite a noisy place. When I say noisy, I don't necessarily mean sound, I just mean there's loads of stuff going on. But in the example we're going to use, we are going to use sound. Of course, we're going to use the example of birdsong. Now, signal detection theory is very useful to help us design experiments and interpret results that we give animals. And this birdsong example is very good at explaining that. Now, let's make an assumption that birds with fast songs, warble really fast, they like all that, they are the fitter birds, okay? So the males that produce these songs are the fitter ones, and the ones with slower songs, like like that, they are less fit. And then we can look at whether a particular signal was detected or not. So there are 4 possible outcomes here. If a signal was given and it was detected by the bird, then that's a hit. Okay? So that's called a hit. If a signal was given but it wasn't detected by another bird, that's a miss. Okay? If there was no signal at all, but a bird detected it, then he must have made a mistake. So that's a false alarm. Okay? And finally, if there was no signal and no detection, then that, of course, is a correct rejection. And what you can do then is plot distributions of these 2 different song types within a population. So at the bottom here we've got song speed, and so that's increasing along the x-axis, and on the y-axis we've got this weird thing, PW health, and that is basically the probability of a particular group of males producing a call at that particular speed. Okay, so this normal distribution curve here is talking about the ones with the slower calls, and the ones, this normal distribution curve here is ones with the faster calls, the okay. Now you might be wondering what this WC stands for. No, it's not the toilet. This is the threshold of acceptance. So that means that anything above this speed will be detected by another male or by a female. Okay? So, as you can see, with this fast song speed, there's some individuals which fall behind this WC. Okay? So they're the misses. So that's where a signal was released but not detected. When looking at this normal distribution curve here, you can see that there are some individuals which fall in front of the WC, the threshold of acceptance. So although they are a less fit individual, they are accepted. Okay? The signal is detected. So these are the false alarms. Okay? Anything here is a correct, is a hit, and anything here is a correct rejection. Now it's this overlap here which is what's creating the noise, it's what's creating the confusion. Okay, so the less overlap there is between these 2 different signals, the greater the difference is, and the easier it is to separate the 2 out. If there's basically complete overlap between these normal distribution curves, then you can imagine it's very difficult to tell apart who is a fit bird and who is a poor, malnourished bird. So the less overlap there is between 2 signals, the better the signal is. But remember, these distributions can change over time. Remember what we were talking about with our visual perception in seeing bright colours, for example. That changes over time. Okay? There are other experiments you can do by changing the intensity of a stimulus, and a great example is when looking at the lateral line system in fish. Many of you probably know that fish have a sixth sense. They have these pits, not quite pits, but like sort of indents around their bodies, which inside contain neuromasts which detect water currents, changes in water movements, and that's very useful for a fish for all sorts of reasons, but we don't need to go into that. So Kuhn et al. Studied the mottled sculpin. God knows why. I'm sure there was a much more exciting fish they could have chosen, but you know, whatever floats your boat. So what they did, they tested not only its lateral perception, but also its frontal perception. Okay, so what they did, they introduced a vibrating stimulus. Okay, so a hit was when the stimulus was vibrating and the fish swam towards it. Okay, because probably thought it was some prey item or something. A false alarm was when the vibrator, or whatever it was standing still, but the fish still swam towards it. Okay, and well, I think you can pretty much guess what the miss and correct rejections were. And what they found was that the number of hits decreased the further away the stimulus was from the fish, and also there were less false alarms when the stimulus was on the lateral side of the body compared to the frontal side. Okay, so quite interesting experiments there. So another way of studying perception is by doing things called psychophysical experiments. Now this is all about learned behaviour. Okay, now a good example is that you can train a bee to extend its proboscis, that's the thing it feeds with. You can get it to extend its proboscis if you introduce a particular odour, and it will only do it in response to that odour. So you've trained that bee, basically. Okay? Slightly more relevant experiments involve the relationship between bees and the plants that they pollinate, the flowers. Okay? Now we know that bees see in UV, right? What this study was testing is whether their ability to perceive iridescence. Okay, now iridescence is the property of a substance that makes it appear to change colour when you look at it at different angles. Okay, so what they wanted to see was the iridescence of the UV radiation. So the hypothesis was that bees probe at the boundaries of UV absorption, places where UV is absorbed very much, and ones where it isn't so much. Okay, so what they did, a scientist called Whitney and his mates got together, actually I think it was a woman, they got together and they made loads of model flowers. I don't know, they probably called Blue Peter or something, I don't know how they did it, but they made some iridescent and some not. Okay, now the ones that were iridescent, in the middle they were given a nice sugary reward, a nice bit of sucrose. Okay, and ones which weren't iridescent had something called quinine in. Quinine is a compound which you find in tonic water, and it's what makes it bitter, basically. So it's not very nice. So the bees aren't going to enjoy that very much, so that acts as a punishment. So what they wanted to see is whether, well, they got a load of naive bees, released them on these flowers, and they wanted to see whether they made an increasing amount of correct decisions. So whether they went to an increasing amount of iridescent flowers, because that would point at the fact that they can detect iridescence. Okay? And what did they find? Yes, Whitney was absolutely correct, that is what happened. So over time, they made an increasing amount of correct decisions. So this is a psychophysical experiment based on learned behaviour. Now when we're doing these sort of things, we must take into account sensory bias. Now what's sensory bias? Sensory bias is the innate ability to detect a signal. Okay? You just do, basically, you can't help it. A good example is in no-list lizards, okay? And their behaviour of head nodding in males. Now, females really like a bit of head nodding action. They absolutely love it. But when you look at the evolution of this behaviour, that isn't actually the reason why head nodding evolved. It was actually involved in male-male competition and defending territories, etc. Another example is in the orange bellies of guppies. So when looking at sensory bias, there are two predictions we can make. One is that the preference to have this particular trait, so the head nodding for example, can evolve before the sensory function evolves. Also, the preference may have another function in another context, okay? And I think we've explained that quite well by these examples. But to emphasise it even further, another study was done by Egger et al., God, what a name, on cichlid fish. Now we know cichlid fish are a great species to study. And this relates into what we were talking about in the last video, when looking at the comparative approach. Okay, so these cichlids have orange spots on their anal fins. And these are displayed to females during courtship. The female then lays her eggs and keeps the eggs in her mouth where the male fertilises them. Nice. Now, in this study it was shown that there was already a sensory bias towards orange spots on the anal fins before it actually became a sexually selected trait. So what they did was, is that they looked at the preference of these fish to different colours, and then they built a phylogeny from it. We learnt all about phylogenies in the evolution series. And what they found was that there was a parsimonious ancestral preference for a sort of reddish colour, okay, on the anal fins. And then loads of different species evolved from that, with different colours. But that colour, the anal fin, was always there in the ancestor. Right, now pay attention, because we're going to now be talking about attention. Now, we need a good definition. Now, you probably know what attention is. Of course you know what attention is, you're not an idiot. But we need to look at it from an ethologist's perspective. So, the definition of attention is the act of concentrating on any one set of objects or thoughts. Okay? And a good way to test for attention or attentive abilities is to do visual searches with increasing complexity, and you can find a load of things online to test your own attention. What you find is that reaction time, measuring reaction time to certain puzzles if you like, is a good way of measuring attention. What we tend to find in general, not surprisingly, is that conjunctives tend to take longer to work out than just single features, and also positives are quicker to work out than negatives. Okay? What I mean by that is that it's easier to detect a positive result than it is a negative result. So let's say you're trying to find a word in a word search, but the word isn't actually there. Obviously that's going to take longer than if the word actually was there. Okay? It's pretty obvious, really, isn't it? So, what are the advantages of attention, evolutionary speaking? Well, it makes you better chance of avoiding predators, easier to find food. There are loads of good reasons. But anything has its costs as well. It may mean you spend longer making decisions, which could make you more at risk to predation if you're not focusing on avoiding predation itself, if you know what I mean. Now, going back to our bees, because we love bees, an experiment was done by Dyer and Chitka to measure their attentive abilities. So what they did, they got a load of flowers that were similar in colour and ones which were different. And what they did, they measured the amount of time it took them to make the decision and the number of correct choices they made. So the ones with the rewards, okay? So the sucrose. And what they found was that with an increasing amount of time, there was an increasing amount of correct decisions. So the more attentive they were, the more rewards that they got. So when the bees spent less time making a decision, they were less good at distinguishing colour, because it was the flowers with similar colours that had the rewards. Another useful thing to do is to develop a search image for a particular stimulus. This was studied in blue jays, which feed on moths. And what they were done is that the jays were trained to develop a search image for a moth, okay? And this could be either a black moth or a white moth. So basically, the jays were trained to peck the moth when it was shown on a screen, and if the moth wasn't shown on the screen, they were trained to press a button. So it would move the next slide along basically, and it would just carry on. Okay, and they were very good at doing this. What they found was, if two moths were shown together, so like a black and a white moth, then they didn't do so well. And one thing I should have said is that the jays were rewarded if they made the correct decision. So that was their incentive, if you like, to keep going. So what all this shows is that the jays were good at developing one search image, but not more than one. This is because the reaction time of those jays slowed down. And what they did another experiment. They took 240 moths on a slide and got the jays to peck at them. Okay? And then what they did with the moths that they didn't peck, they created similar morphs, another 240, based on the ones that they didn't peck, which they showed the jays the next day. Once again, the moths that they couldn't find that day were taken and used to create similar morphs, and they found that by the end of the experiment, it was only the most cryptic morphs that remained. So this shows that developing a search image not only benefits the predator, but it also is benefiting the species because it's allowing for the evolution of more cryptic forms, so they're less likely to be seen by a predator. But anyway, that's all I've got time for today. I must say I'm weirdly tired today, but I hope that was okay for you. Please let me know if I made a mistake. I might have done at some point. I'll say that was the hardest lecture I've had to do so far. But thank you very much. I'll be back tomorrow when we'll be learning about learning. Great. See ya.